And without further ado, it's going to begin with abstract number 101. Ratna Chaturvedi, who's a postdoc in the Lee lab, will be telling us about the work she's done on the function of a glial syncytium in the Drosophila visual system.
Thank you very much. And first of all, I would like to thank organizers for selecting my work for the presentation. Okay. So I'm going to talk about Glial network recycles the neurotransmitter histamine in Drosophila visual system. Vision is an important sensory organ system in all the animals, including vertebrates and invertebrates. 95% of the information what we perceive from outside world is through our visual system. Therefore, continuous signaling across this, in this visual system is important. So first sensory cells in the visual system is called photoreceptors, and these are light sensitive cells which continuously send signals to the downstream neurons. Both in case of Drosophila and vertebrates, these two photoreceptor cells are highly active cells, and they requires large amount of neurotransmitters for the continuous signaling. In case of mammals, including humans, this is glutamate, while in case of Drosophila, it is histamine. So to fulfill the large requirement of neurotransmitters in the photoreceptors, there are two possible methods. One, it could be a de novo synthesis, which is less energy efficient and the slow process. Other method is recycling method, which is more energy efficient, faster, and dominating pathway because it reutilizes the already synthesized neurotransmitters. So here comes the role of glia, which plays very important role in this recycling mechanism, because it takes up all the used or neurotransmitters present in the synaptic cleft, and then it recycles back to the photoreceptors. So the role of glia in this recycling is well known, but how do glia deliver neurotransmitters back to photoreceptors is still unclear. So we are addressing this problem, and in Drosophila visual system, Drosophila visual system has two compound eyes. Each compound eyes are made up of roughly 800 ommatidiums. And out of these each ommatidium is made up of 8 photoreceptors. Out of these 8 photoreceptors, 6 photoreceptors forms a synapse in the first optic neuropil, which is called lamina. So today I'm going to focus on the synapse between the photoreceptors and the lamina neurons and the recycling of histamine here. So if you look at the detailed mechanism of histamine recycling, glia take up the histamine after it binds to the receptor and release back, and then it converts into the carcinin, which is the inactive form, by conjugating histamine and beta-alanine together with the presence of an enzyme called ebony. And this carcinin is then transported back to the photoreceptors, where in the presence of another enzyme, which is called TAN, is dissociated back into the beta-alanine and histamine. And this free histamine goes back to the synaptic vesicles. So if you look at the anatomy of the lamina, there are multiple layers of the glia present in the lamina. This is the representative figure. Here, this is the lamina, and this part is retina. So if you look at the different layers of lamina, you can classify it as marginal glia, epithelial glia, satellite glia, pseudocartilage glia, and fenestrated glia. So based on the immunostaining, on the ebony enzyme is only present in the epithelial glia. So it is expected, it was assumed that this recycling is limited or confined to this region, which is the neuropil region of the lamina, and this is mostly occupied by epithelial glia. So if this is true, then most of the carcinin and beta-alanine should be present in the epithelial glia region, which is neuropil region, and it should not be present in the pigment cell region or retina part. So to confirm this, we stain carcinin and beta-allelin, and this is the cross section of the retina, and this is the lateral view of the retina, co-stained with the choptin, which is a marker for the rhabdomeres. So you can see here that there are a lot of staining for carcinin and beta-allelin, which suggests that these metabolites of histamine recycling pathway are actually present in the retina. So to confirm that which type of cells they are, we co-stain with ATPase alpha, which is actually present in the plasma membrane of the photoreceptors and the pigment cell, which surrounds the photoreceptor cell body. And whatever the signal present or co-level with this marker is actually supposed to be present in the pigment cell. And the signals which are present not co-level with this marker is supposed to be present in the photoreceptor cell body. So you can see this is the TAN mutant and this is ebony mutant, which has large amount of carcinin and beta-allelin. You can see that large amount of signals are present actually in the pigment cells. So based on this, we can say that histamine recycling may not be limited to the lamina epithelial glia. So next question what we ask is, how is carcinin transported to pigment cells from the epithelial glia? To answer this question, we look at the possibilities, because this epithelial glia is not directly connected to the for pigment cells. So there might be there is multiple layers of other types of glia present here. So it could be a glial network, and there might be a lot of transporters here which are transporting these molecules across this glial network. So first we look at gap junctions, because it is already known that a lot of gap junctions are present in the glia and they form a network. So we screen it for using RNAi knockdown, and we knock down using all inaxin proteins which are present in the Drosophila using different Gal4 lines, and we found that inaxin 2 could be one of the candidate, because when we knock down inaxin 2 in different lines, we found that using DPO-Gal4, which is a glia-specific driver, we found the lethality, which can be rescued by overexpressing inaxin 2 under DPO-Gal4 driver. So this suggests that inaxin 2 is essential for the glial function. So next we look at whether this inaxin 2 is also present in the lamina glia. For this we stain inaxin 2 in the lamina, and we found this signal in the glia, and this DLG is the marker for the neuropil, which suggests this is the epithelial glia region. So you can see that a lot of inaxin 2 are present in the lamina. To confirm that this is actually glia region, this is present in the glia, we co-level it with the RePo-Gal4 driving GFP, and we found that GFP is co-localized with the inaxin 2. So next we look at the if we can knock down this in a specific if we can knock down this in the satellite glia, which is a specific subtype of glia and which is present between the pigment cell and the epithelial glia, what will be the result? And then we knock down this in the satellite glia and look at the carcinoid distribution pattern, and we found that there was a reduction in case of a knockdown flies as compared to the wild type. This could be the two possible reasons. Either carcinin cannot be transported to the pigment cell, that's why it is not able to get the signal here. But sorry. There is some, I don't know. Oh.
No worries.
Oh, sorry. It's okay. So next we asked that if this is the case, then you should see the lot of carcinogen accumulation in the lamina. In contrast to that, we found that there was a decrease in the carcinogen in lamina area also. So that suggests that there is somehow knocking down the annexin-2 satellite glia is affecting the synthesis of carcinogen. So there could be another possibility because carcinogen can be synthesized by conjugating with beta-alanine and the histamine. It is possible that histamine is actually not able to transport it back from the photoreceptor and pigment cell to the epithelial glia. That's why carcinogen is not able to synthesize. To know this, we look at the knockdown flies and the beta-alanine distribution pattern, and we found that in this case, knockdown, you can see the large amount of beta-alanine is accumulated in the pigment cell as compared to the wild type, and then we look at the epithelial glia region, which is co-stained with this TLG, and this mark the reason for the epithelial glia. And you can see in this case that there is a reduction of beta-alanine, which suggests that somehow blocking this network is blocking the transport of beta-alanine from pigment cells to the lamina epithelial glia. So as a consequence, you can see that in case of knockdown flies, there is a reduction of histamine in this knockdown flies. Sometimes you can see some accumulation in the epithelial glia region. So based on these results, we can conclude 3 points: that histamine can recycle through the long distance pathway that involves both the lamina and the retina. Carcinogen is transported from the lamina into the retina pigment cells, and beta-alanine is transported from the retina into the lamina epithelial glia. So next we asked that what is the impact of disrupted long distance histamine recycling on visual synaptic transmission. And for this, we use the one tool called ARG, where you can see here on and off transients. These two spikes are actually representing the transmission from photoreceptor to the postsynaptic lamina neurons. And if you look at the knockdown flies, you see that these on and off transients are absent, which suggests that these are somehow having defects in the visual transmission. And this is consistent with the other histamine recycling mutants, which confirms that there is this knockdown fly is actually defective in the visual transmission. This is the quantification. Then we asked that what is the impact of disrupted long distance histamine recycling on the visual behavior. For this, we have developed one assay in the lab, which is called visual alert assay. In this assay, we allow a single fly to move upwards, and then we challenge this with the moving box across his visual field and record its response. So in case of a wild type fly, fly shows a freezing response as this block moves towards it. While in case of knockdown or visual defective flies, these flies are actually not responding as wild type freezing response. So based on this, we calculated and quantified this behavior, and we found that these knockdown flies are showing similar type of behavior abnormalities as other histamine recycling mutants are showing. So based on this, I have concluded that when histamine is taken up by the epithelial glia, it can be conjugated with beta-alanine and synthesize carcinogen in presence of ebony, because ebony is only present in the epithelial glia. Then it transported through the entire glia network and then reaches to the pigment cell, and then it goes back to the photoreceptor cell body, where in presence of another enzyme TAN, it can be dissociated into the histamine and beta-alanine, and then this histamine can go to the synaptic vesicle for the next release. And here the free beta-alanine comes back to the pigment cells, and then through the entire glia network, it reaches to the epithelial glia region, and where it is ready to synthesize the carcinogen. With this, I would like to thank my lab members, my PI Hongsheng, Keith, Pei, Luan, Caitlin, and Cassie, Department of Neurobiology, UMass Medical School, Mark Freeman Lab for the Calvo lines and all the help, NIH funding, and Dr. Reynard Hart for inaxin 2 antibodies, and US inaxin 5. Thank you.